Here for Kate. Everybody is excited about bagels and coffee. Thank you, Maddie, for arranging those. Okay, so I would love to introduce Kate Sinnott today. I remember the first email I got from Kate. So I received an email from a student who had a linguistics degree from Macalester College. And I thought, well, that's interesting, applying to work in a lab that works with wetlands, plants, and restoration. But I thought, hey, she probably can write and is a very clear thinker. I thought, okay, this could work. I looked a little bit further down her resume, and she had also taken a bunch of ecology and restoration coursework, and she was working at the Tallgrass Prairie Center. And she mentioned that she had worked on propagating 30 different prairie species, and she loved sedges. That was what did it for me. For those of you that don't appreciate that joke, I spent 6.5 years studying sedges as part of my PhD. So Kate knew how to win me over. So I was intrigued. So I received her email in February of 2020. Do you remember February of 2020? It was a very interesting time. So we had a few conversations. I thought, oh, great, let's schedule a visit. Then a global pandemic hit. So Kate is the first student that I brought on that I had not ever met in person. I was a little bit nervous about that, but we set up a full day Zoom campus visit. I was looking back at over my emails at the from that time, and there was instructions to the students. If you have never used Zoom before, this is how you download it and use it. I think we had no idea what was about to hit, but you know, it's kind of a miracle that it all worked out. I had just received funding for a brand new research area for my lab in aquatic plant restoration. And I knew that the student coming on this project would need to be highly curious, very creative, have a great work ethic, and be able to work independently. We just didn't know much at all about aquatic plants in this region. We couldn't even identify most of these species. We didn't know where they were growing. We didn't know how to collect them, how to propagate them. So luckily, Kate turned out to be the perfect student for this project, with a passion for plants and propagation and restoration. And she dove right in and chartered a new path. So she's developed this amazing research infrastructure in aquatic plants, and that's really set my lab up for, you know, the years to come. So it's been a phenomenal effort on her part. And already she's greatly enhanced the capacity for aquatic plant restoration in the region. And because of her accomplishments, I'll just embarrass her a little bit more. She was awarded the Honey College of Natural Resources Masters Student Researcher of the Year this year. And I'm excited to turn it over to Kate here to talk about her work in aquatic plant restoration. So welcome, Kate.
I'll put it back. Yeah, thank you. Thank you so much, Karin, for that introduction. And I also wanted to give a quick thank you to Maddie for organizing the snacks today, and for Alana for fielding all the tech questions. I'm really excited to see so many familiar faces in the audience and so many familiar names on Zoom to share with you my master's research on overcoming barriers to aquatic plant restoration, addressing gaps in species identification, and planting techniques in the Intermountain West. So this is where we're going in this presentation. We'll be talking about what are aquatic plants and why do we care about them? Where is this research taking place? What are the barriers to aquatic plant restoration in the Intermountain West? And how we address those barriers in chapters 1 and 2 of my thesis. So what are aquatic plants? I wanted to be really explicit in this definition because people use a lot of different terms for wetland plants that overlap in different ways. So we are not talking about emergent plants, but we are focusing on floating leaf species, free floating species, and submerged species. And I also wanted to make this distinction, like Karin mentioned, because in the past, the Wetland Ecology and Restoration Lab has focused on emergent plants. And so this restoration project really heralded in a new research program, and that affected a lot of the questions that we were asking. So why do we care about aquatic plants? They have been tied to many different ecosystem functions, including primary production, nutrient cycling, hydrologic regime, and habitat for different aquatic species. And these functions then tie into critical ecosystem services like provisioning of drinking water or food, and habitat for culturally important species. But despite this importance, wetlands, particularly in the Intermountain West, have been degraded over the past 150 years or so by different threats, such as eutrophication, land conversion, species introductions, drought, and climate change, just to sum it all up. So now feels like a good time to revisit question 1 of the quiz that I had up right when you walked in. So I'll read the question, then I'm going to ask what you think the answer is. Quantitative global assessments of aquatic vegetation dynamics show that submerged aquatic vegetation is, so raise your hand if you think A, increasing worldwide, B, staying about the same, or C, decreasing. Okay, that tracks with the people. What I know about the people in this room, it is decreasing. And not only is aquatic vegetation decreasing worldwide, but it's an accelerated decline. So it is declining now more than ever. But the good news is that restoring aquatic plant communities can help reestablish some of those ecosystem functions and services. But one of the questions I've got a lot over the last few years is why should we restore it? Why should we actively restore aquatic plants? After all, if you dig a stream channel or you dig a pond, plants are going to come in. And that's true to an extent, but it's analogous to a terrestrial system in which if there's a large disturbance on the landscape, plants will come in. But are they plants that address specific restoration goals? Do they support the different ecosystem functions and services that we want to reestablish? Likely not. And so actively restoring and floating plants allows us to address specific restoration goals. For example, invasion resistance, limiting sediment resuspension if you want to improve water clarity, or creating habitat for aquatic species like waterfowl or a specific fish species. So speaking of a specific fish species, let's zoom in on the site we're focusing on. So we are looking here at Utah Lake, which is about 40 miles south of the Great Salt Lake. And it's a large freshwater lake that has been affected greatly over the past 150 years or so by many of those anthropogenic stressors that we discussed earlier. And one of the near casualties of those stressors has been the June sucker, which is a fish species that is endemic to Utah Lake. And it's been affected by many of those stressors, but one of them has been a loss of critical breeding habitat. So a little bit about the June sucker. They travel up the tributaries to the lake to spawn, and then the larvae kind of hang out in those tributaries until they're large enough to live in the lake itself. But as those tributaries were channelized, that water became faster, it became deeper, it became colder, and a lot of the area and plant material for that critical breeding habitat was lost. So the June sucker had been listed as endangered in 1986, and that was lessened to threatened just a few years ago, but that's largely due to stocking efforts. We don't see these really large, naturally repopulating populations of June sucker. So one of the ways we could address that is by reestablishing that critical breeding habitat. And we'll zoom in on a project that is doing just that. This is, of course, the Provo River Delta Restoration Project, which is located at the confluence of the Provo River and Utah Lake. And the main goal of the project is to restore the June sucker, and in doing so, also restore the natural ecosystem of the river delta. And they're doing this by diverting most of the flow from the channelized Provo River into the restoration site, where they have excavated a series of braided channels and ponds. But when they were soliciting bids for aquatic plant material to populate these newly excavated ponds and channels, they did not receive any bids. And this is because native plant producers are not producing these plants in our region, and restoration practitioners aren't used to using them. So instead of putting that money into purchasing plant material, they invested it into research to increase capacity for aquatic plant restoration in the Intermountain West, and as that applies to the river delta. And I want to give a shout out here to Melissa Stamp, who's the project coordinator of the Provo River Delta Restoration Project with the Utah Reclamation, Mitigation, and Conservation Commission, because Melissa really saw this need and was the catalyst for this project to begin. Yeah, Melissa. So as part of that research, we first needed to identify the different barriers to aquatic plant restoration in our region. We wanted to know why producers aren't growing these species and why practitioners aren't using them. So we did this in a few different ways. We reviewed the literature, both peer-reviewed and gray literature. We had conversations with wetland professionals, including restoration practitioners, wetland managers, and botanists. And these were not like formal IRB interviews. These were informal conversations that we tried to have with a lot of people. And we also had consultations with aquatic plant specialists. And here I'd like to give a shout out to Casey Williams of BioWest down in Texas, who was instrumental in getting us started. So here are the barriers that we identified. First, a lack of confidence in species identification amongst wetland professionals. Second, that planting methods for aquatic plants are variably successful and not scalable over large restorations. And that the impacts of planting designs are largely unknown. And so we addressed each of these barriers in my thesis. So barrier 1 was addressed in chapter 1, and barriers 2 and 3 are addressed in chapter 2. So let's dig in and talk about chapter 1. So proper species identification is critical to the management and restoration, not only of aquatic species, but really any species. In order to adequately conserve or restore these species, you need to understand what is around you in the ecosystem. However, aquatic plants in particular can be challenging to identify for a few different reasons. First, that they are at least partially underwater, so you have to really get in there to look at them and identify them. But also, many of the species have shared leaf characteristics and reduced flowers, so it can be difficult to tell these plants apart. And the flowers aren't big and showy, so you really sometimes have to even get under a scope to identify them. Case in point, these are all different species. And when I started, I was not familiar with them either. And so it was kind of daunting, but I really believed what we needed was a resource that allowed us to tell the differences, that picked out the differences between these look-alike species and was accessible in terms of information and physical accessibility. So we started compiling information. Again, I was not familiar with these species at first, so my first stop was the Intermountain Herbarium here at USU, which is a great resource. And it was very helpful to look at these physical specimens of the species, as well as on the vouchers. There's a description about the habitat in which it was found, so we were able to familiarize ourselves with the different habitat of individual species. We also use many different books, starting with the Wetland Plants of the Great Salt Lake book by Becca Downard et al. And, but however, many of the guides that were specific to aquatic plants came from farther afield. So the Aquatic Plants of the Upper Midwest is an amazing aquatic plant guide, but it is not Utah, right? So there's a lot of information in there that does not apply. We also use books like Utah Flora and the Aquatic and Wetland Plants of the Southeastern and Southwestern United States. So much information here, but they are hefty books, right? So you really have to examine them to find the information that you're looking for, and it's not like you are going to bring a guide like that into the field. We also used a lot of databases to get an idea of the range of these species, where people are finding them on the landscape today, as well as additional information. And last but not least, we used a lot of observation. So we were able to travel to wetlands throughout Utah and observe these plants in their habitat, take samples of them, bring them back to the lab, and put them under the scope and just really look at them and start to identify them and learn the different tips and tricks that allowed us to tell them apart. So I'll give you a couple examples of those tips and tricks. This is Xanakelia palustris, and right at the beginning, we brought a sample back to our lab, and one of our technicians at the time, Amanda Mast, said the seeds on this species look like bananas. And that became a distinguishing characteristic of that species for us. Does this plant have seeds that look like bananas? Or this plant, Ruppia cirrhosa, it's very easy to distinguish when it's fully in flower, but when it's not in flower, we had to develop other ways of identifying it. And so after walking around a wetland and looking at a bunch of plants in the field, we observed that when the inflorescence is coiled inside the leaf sheath, it looks just like a stack of burgers. So that became the distinguishing characteristic for this species at this particular life stage, a stack of burgers. So we combined all of that information into the creation of the Floating and Submerged Plants of Utah Pocket Field Guide. And I have a physical copy here that I'll pass around during the questions. So we have 36 species included in this guide, and I'm just going to go through one of the pages to show you the kind of information that we have here. So this is Xanachellia palustris that we were looking at before. We have the family name, the species name, and the common name, followed by characteristics of its habitat, stems and roots, leaves, flowers and seeds, and an additional facts section where we put synonyms or ways to tell it apart from lookalike species, as well as fun facts. At the bottom, we have the nativity of the species, its duration, so if it's annual or perennial, and also its commonness, and these are specific to the state of Utah. And we also incorporated our little tips and tricks. So here we have that the seeds are banana shaped. Or on this page of Ruppia cirrhosa, we have a little bubble that says the inflorescence looks like a stack of burgers before it emerges from the leaf sheath. And I know that these are silly, like little tricks, but they really do work, and they really got at what we were trying to do by making the content in this guide accessible to whoever wants to use it. So in my mind, Ruppia burgers, Ruppia burgers. So now let's put that knowledge to the test. What aquatic plant is this? And you can just shout out the genus. Yes, thank you. It is Ruppia cirrhosa. So again, silly, but you just proved to me that it worked. So thank you. We not only included native species, but we also included many species of aquatic invasive species, including ones that haven't been found widely in Utah yet, or haven't even been found in Utah at all, because we want people to be able to identify them and contact and report them to their county weed supervisor as needed. So remember, these plants that are all different species that look pretty much the same, they are all from the Pondweed family, Potamogeton. And this is a particularly tricky family because of the shared leaf characteristics and reduced flowers. So we created a pondweed key that is specific to the members of the pondweed family that are included in this book. And this is a dichotomous key that allows users to use its physical characteristics to ID it to species. In addition to wanting this guide to be accessible in terms of content, we wanted it to be physically accessible. So this guide is pocket-sized, it's 4 by 6 inches. The pages are waterproof and tear-proof. And it is ready to go in the bottom of your kayak or in the pocket of your waders to take out with you on your research or recreation this summer. And this guide is currently in the process of publication through extension here at USU. So stay tuned, hopefully this summer, to see it out in the world. So to recap on our barriers here, we addressed this lack of confidence in species identification through the creation of the Floating and Submerged Plants of Utah Pocket Field Guide. And now let's dig into barriers 2 and 3 on planting techniques. So this is the whole title of this chapter, Restoration Techniques to Enhance Plant Establishment and Project Feasibility in Aquatic Ecosystems, but we will call it planting techniques for short. So decision-making plays a crucial role in restoration, with practitioners balancing ecological, practical, and financial considerations to make decisions about different revegetation techniques, including plant material type, so the form in which the plant arrives at the site. So this can be a seed or a plant or a fragment of the plant, the planting method, how the plants are introduced to the site, and planting design, or the arrangement of plants within a site. And we're going to go a little bit more in depth on each of these concepts. Aquatic plants reproduce and spread in many different ways, including via seed, via stem or root fragment, or by bud. But many aquatic plants are known to regenerate primarily vegetatively. And so when we're deciding what type of plant material to use in restoration of aquatic species, it makes sense to look at vegetative propagules. So here we're going to look at two of those. The first is stem fragment, which is exactly what it sounds like. It's a bit of the stem of the plant, which may have leaves on it, or at least a leaf bud, so it has some meristematic tissue. And these stem fragments are introduced directly to the restoration site like that. Or you could take it a step further and take the stem fragment, plant it into a cube of soil or a plug, and allow it to grow out. So it grows roots into the soil. And then you transplant that entire plant into the restoration site. But both of these options have trade-offs. So a plug is just a more mature plant, but that soil around its root ball can reduce transplant shock, and it also provides initial fertilization to the plant upon introduction. However, growing plants and transporting them and planting them at the site can require a significant amount of infrastructure. Stem fragments, on the other hand, require a lot less input to produce and transport, and aquatic plants have been shown to be uniquely adept at recolonizing disturbed areas via stem fragments. However, the efficacy of stem fragments in aquatic plant restoration, particularly in inland aquatic systems, has not been shown in a research context. So we don't really know the risks of using stem fragments as the primary propagule. Moving on to planting methods, there are a myriad of ways to introduce plants to a site. The two we'll focus on today are hand planting and anchoring. So hand planting is when the plant itself is placed into the substrate by hand. And anchoring is when there is weight added to the plant, which allows it to be dropped from the surface of the water and then anchor onto the substrate. But again, both of these options have trade-offs. So hand planting the plants results in a high level of plant soil contact, which has been shown in seagrass restorations to improve restoration success. However, hand planting can be very labor intensive, expensive, or potentially infeasible in sites with low visibility or where safety is concerned for practitioners. For anchoring, this is a method that is much more scalable across large systems because we are able to drop them from the surface of the water, eliminating the need for hand planting. And though there is a lesser level of plant soil contact compared with hand planting, because the plant is anchored to the substrate, you still do see moderate levels of that contact. However, these methods are still being developed and they're still very much in a proof of concept phase where we're understanding exactly the techniques that work or don't work. For planting design, in wetland restoration, it has been heavily influenced by forestry science, which emphasizes the minimization of competition between individuals through a dispersed planting design. But that does not incorporate a robust body of research showing that planting the plants in a clumped planting design may promote positive interactions between individuals. And this has mostly been shown in seagrass and coastal marsh communities where these clumped plantings ameliorate the physical stress of wave action. So one of the stressors that we identified in the context of the Provo River Delta that could be ameliorated by this clump design is anoxia. So we anticipated that the water at the site would be very still, no mixing. This was prior to when it was connected to the river of the lake. So very still water with anoxic conditions at the substrate level. Aquatic plants have been shown to have leaky roots where if one individual has excess oxygen, it will leak that oxygen into the substrate, which a neighboring individual could then take up from the substrate. So this is one way a clumped planting design could improve restoration success at the Provo River Delta. So now that we have a handle on those concepts, let's talk research questions. We asked, what are the best planting methods for plugs and fragments to maximize success? We predicted that the planting methods that resulted in increased soil-plant contact would lead to the highest cover. Second, we asked, does manipulating planting design affect restoration success? And we predicted that a clumped planting design would promote positive interactions between plants and increase establishment. We evaluated these questions in 2 field experiments. Each field experiment focused on a different plant material type. So experiment 1 focused on plugs and experiment 2 focused on fragments. So we're going to look at the methods for each of these experiments. For experiment 1, again, we focused on plugs as a plant material type. And these are the species we used in this experiment: Stuckenia pectinata, Ruppia cirrhosa, stack of burgers, and Potamogeton nodosus. And these species were selected because we were limited to a list of 9 species that had been identified in the original revegetation plan for the Provo River Delta. And from that list of 9 species, we selected 3 that root in the substrate, are relatively common in the area, and span a wide range of growth forms and habitat preferences. We have 3 planting methods here: coir pellets, burlap wraps, and hand planting. So coir pellets and burlap wraps are both anchoring techniques, which are intended to be more scalable because they can be dropped from the surface of the water. So we're comparing these here against hand planting. And I wanted to take a moment to talk about how these different planting methods were produced. So for coir pellets, the stem fragment was planted into a pellet that's lined in mesh and then allowed to mature and introduced to the site like that. For burlap wraps, the stem fragment was planted into a cube of soil or a plug, grown to maturity, and then at maturity, it was wrapped in burlap and tied with twine. And lastly, hand planting, the stem fragment was planted into the cube of soil and then planted directly into the site with no additional material added. We also looked at 2 different planting designs in this experiment: the clumped planting design with all of the plants touching, and a dispersed planting design where they are spaced 50 centimeters apart. And to connect this back to our predictions, we predicted the planting method that resulted in the highest plant soil contact would have the highest establishment, so that would be hand planting. And for plugs, we predicted that a clumped planting design would ameliorate the stress of anoxia and result in higher establishment. So revisiting our quiz question to make sure this is clear: coir pellets and burlap wraps are, so raise your hand if you think it's A. Okay, raise your hand if you think it's B. And lastly for C. Okay, I tricked you here. So most of you are right. It is both. It is both anchoring planting methods and more scalable. So these anchoring planting methods, again, can be dropped from the surface of the water, making them more scalable than hand planting. I also wanted to take a moment here to talk about where we sourced our plant material from, because this was a barrier for us getting started, and we know it's a barrier for restoration practitioners. We don't have large populations of many of these species that we can go back to and harvest plant material from for use in our research. So we built these tanks at the Millville Aquatic Research Facility. The tanks were there, but we lined them with pond liner, filled them with soil and water, and built an irrigation system. This is for 30 of the tanks. And then we went to wetlands throughout Utah and collected populations of our target species. And so on the left, this is plant material that had just been introduced to the tank. And on the right is the next season. So this is when the population is established, and all that green is nice new growth. And these are intended to be foundation population tanks, which exist somewhat indefinitely, that we can go back to and harvest plant material from for use in our research and future revegetation efforts. So they're kind of like banks for these species. So for to grow out our plugs, we harvested plant material from our foundation population tanks, planted them in their respective plugs. And the plants were considered ready when roots were protruding out of the bottom of at least 50% of the plugs in each tray. This experiment took place in a pond at the river delta that had been excavated in 2021. We used a factorial design, so all of those variables were present in every combination. We had 4 blocks in the pond, and each block contained 18 1 meter squared single species plots, and the plants were introduced at a density of 9 plugs per plot. Because the burlap wraps and coir pellets, the anchoring planting methods, were not affixed to the substrate, we still wanted to enforce the planting design to assist between treatments. So we affixed those treatments to a wire frame. So here you see Nate tying some burlap wraps onto one of the wire frames. And then we affixed those to the substrate or hand planted them in the water. And throughout the monitoring period, depths in this pond were mostly below 80 centimeters or so. For monitoring, we collected visual estimates of percent cover as a proxy for establishment success. And data was collected approximately bi-weekly for a little over a year. We collected these visual estimates either using a bathoscope, which I'm holding in that photo. It is just a 5-gallon bucket with the bottom cut off and some plexiglass taped on there. But this allowed us to see what was going on under the water a little better. Or we also did this by eye if conditions allowed. And lastly, we collected environmental data on water depth, temperature, and passively recolonizing species. And to my relief and delight, they grew. And I just put these photos in because I'm proud of them. And so on the left here, these are all from our plots. This is Potamogeton nodosus on the left, Stuckenia pectinata in the middle, and Ruppia cirrhosa on the right. Moving on to experiment 2 methods. Again, this experiment used fragments as the plant material type. We used the same species as the previous experiment. And because we were not affixing these plants to the substrate, we used different planting methods. First, mesh bags in which the fragments were enclosed in a mesh bag, and or compared to loose, which basically means without a mesh bag. We also used clumped and dispersed planting designs, but because we were not affixing these plants to the substrate, these planting designs played out mostly in how the fragments interacted with the planting methods. So for the mesh bag clumped treatment, all of the fragments for a plot were in a single bag. For the mesh bag dispersed, all of the fragments were in separate mesh bags. For the loose clumped, all of the fragments were tied together with a little bit of twine. And for the loose dispersed, they were just loose with no added material. And again, to connect this to our predictions, we thought mesh bags as an anchoring treatment, which would result in increased soil plant contact. So we anticipated this treatment would have higher establishment, and again, a clumped planting design. This experiment took place at the river delta in a channel that had been excavated in 2021. We again used a factorial design. We had 4 blocks. This aerial image is from Google Maps, which is cool. And each block had 12.8 meter squared single species plots. And we introduced the fragments at a density of 9 fragments per plot. Because we weren't affixing these plants to the substrate, we still wanted them to stay in the plot so we could see what was going on. So we built these frames that were lined vertically with polyethylene mesh. And this is Karin modeling one of them prior to installation. And this is what they looked like when they were installed in the channel. And so we just floated the plants and their different treatments into the each of those plots. And so here we have a dispersed mesh bag treatment. Again, we collected visual estimates of percent cover as a proxy for establishment success. And we were able to do this by eye because water depth was lower in this experiment, and we did not require the use of a bathoscope. This data was collected from August to October last year. This is a much shorter monitoring period. We were limited a little bit logistically in terms of monitoring length, because at the time of experimentation, the restoration site was not connected to the river or the lake. So we were working here with groundwater. However, it was connected about a little over a month ago. So the hydrology this season is changing a lot. I'm sure that it already has. And so these plots here are all probably very much underwater. So that kind of limited our monitoring period, but we still got a good idea of initial establishment. And we lastly also collected environmental data on water depth, temp, and passively recolonizing species. For analysis, we used generalized linear mixed models to assess the effects of planting method and design on proportion cover. So we transformed percent to proportion cover, and the species were assessed in separate models, and we used the data from the peak date of each respective species. These are the factors that we included in the models. They're basically the same for each experiment. So the fixed effect factors are planting method, planting design, and their interaction, and the random effect factor is block. Let's look at some results. So on this screen, there are 3 plots. Each one is a different species. On the y-axis, we have percent cover, and on the x-axis is date. And this is just to kind of get an idea of how the plants grew over time. And one thing I'd like to point out is that we had low initial percent cover, but it increased rapidly in the second growing season. And you can also kind of get an idea of the differing phenology of each of these species. Looking at model data, so these are model derived means. On the y-axis, we have mean cover proportion, and the x is divided out by species on their respective peak dates. In this plot, we are just looking at planting method, and we found that Potamogeton nodosus and Stuckenia pectinata did not show a significant effect of planting method on their cover. However, planting method did have a significant effect on Ruppia cirrhosa cover here with coir pellets and hand planting performing significantly better than burlap wraps. This plot is laid out the same with mean cover proportion on the Y and the species on the bottom. And here we're just looking at planting design. And for Ruppia cirrhosa and Stuckenia pectinata, we did not see a significant effect of planting design, and we did see a moderate effect of planting design on Potamogeton nodosus cover. Moving into experiment 2, again, this is percent cover over date. And there's a couple things I wanted to point out in these plots. First is that mean percent cover did not exceed 5% throughout the entire monitoring period. And this makes sense with what we saw in those experiment 1 plots where initial percent cover was very low, but we did still get an idea of the initial establishment dynamics. The other thing I wanted to point out was that you can see throughout these plots that there's a bit of a drop that happens in mid-September. And we believe that is due to this, which is showing mean depth of standing water. We see a big drop in mid-September that resulted in some of the plots reaching 0 centimeters of standing water. And I'll discuss this a little bit more later on. Looking at our modeled data, we have mean cover proportion here for each of the species. In this plot, we are just looking at planting method. And again here, we found that planting method did not have a significant effect on the cover for Potamogeton nodosus or Stuckenia pectinata, but it did have a significant effect on Ruppia cirrhosa cover, this time with the loose planting method performing significantly better than mesh bags. Here we are looking at planting design, and we did not see a significant effects of planting design for any of the species in this experiment. So to connect this back to our research questions and predictions, we asked what are the best planting methods for plugs and fragments to maximize success. And we predicted that the planting methods that resulted in increased soil plant contact would lead to the highest cover. And so this would have been hand planting for experiment 1 and mesh bags for experiment 2. However, we did not see significant effects of planting method across both experiments for 2 of the species, Stuckenia pectinata and Potamogeton nodosus. So for experiment 1, this indicates that the scalable planting methods could be viable alternatives to hand planting. And for experiment 2, it indicates that there might not be value added by enclosing the fragments into mesh bags. However, Ruppia cirrhosa acted a little differently. And although we evaluated these species in different models, we can see qualitatively that they responded differently. So Ruppia cirrhosa had significantly higher cover with coir pellets and hand planting than with burlap wraps. But that doesn't mean we just forget about Ruppia cirrhosa, right? We need to consider species identity in the planting method selection in order to maintain restoration success. Question 2, does manipulating planting design affect restoration success? We predicted that the clumped planting design would promote positive interactions between plants and increase establishment. So that's the clump design. But we didn't really see that across experiments and species, with an exception of moderate evidence for Potamogeton nodosus. And we do have a guess as to why the clumped planting design did not respond the way we thought it would. Originally, we had identified anoxia as one of the main stressors due to that lack of mixing. However, there was a lot more movement in the water in the site than we had initially expected. And though we didn't measure oxygen levels, we presume that this meant that anoxia might not have been a stressor here. So this lack of observed effects of planting design may be attributed to the low stress levels at the experimental site. But we did observe something that could lead to a lesson. And that's related to this. So remember that drop in water mid-monitoring period. So that drop in water resulted in some heterogeneity at the plot level, which was derived from the microtopography of the plot, resulting in some areas having no standing water and some areas of the plot having a little bit of standing water. And because of this heterogeneity, the results from this experiment were difficult to conclusively parse, but our qualitative observations showed that a clumped planting method, the fate of all of the plants in that plot was dependent on the one location where that clump ended up. So if it was like here in an area with no standing water, those plants were toast. But with a dispersed planting design, we see a greater likelihood of at least one or two of the plants remaining submerged, which could mean they could survive until, the next rainstorm. So these qualitative observations of the dispersed and clump planting designs offer a bet hedging strategy in situations with dynamic water conditions, which is going to become all the more prevalent due to global climate change. So let's distill all of those lessons that we learned into recommendations for practitioners. We suggest that carefully pairing species identity and scalable planting methods can increase the feasibility of the project by allowing more options while maintaining the success of restoration projects. So Stuckenia, pectinata, and Potamogeton nodosus work with any of the planting methods, which gives the practitioners a lot more options. But again, we don't want to forget about Ruppia cirrhosa. Making sure that these plant communities are diverse is a very important component to making sure that these restorations persist. And so we want to carefully pair these species with the scalable planting methods. Recommendation number 2 is that logistical considerations rather than potential ecological differences can guide planting design choices. By this, we mean that whatever works best for practitioners is what they can do, because we did not see significant ecological differences between the treatments. So connecting this back to our barriers, we addressed barrier 1, the lack of confidence in species identification, through chapter 1's creation of the Floating and Submerged Plants of Utah Pocket Field Guide. And we addressed barriers 2 and 3 on planting methods and designs through field experiments that assess different planting techniques, and hopefully will elucidate the decision-making process for restoration practitioners. Future directions, I don't think this will surprise anyone. Small-scale experimentation is really important to identify the planting methods and designs that work or that don't work, but the next step is certainly scaling those up spatially, so over larger spatial extents, temporally, so longer monitoring periods to really assess the differences, and environmentally, so over larger environmental gradients. Focusing more on my project, we are hoping to release the Pocket Field Guide this summer through USU Extension, so keep an eye out for that. And lastly, I didn't talk much about this in the talk, but an additional barrier that we had identified is that for plant vendors, starting to grow aquatic species can require a lot of infrastructure, and that can be a turnoff. So we are developing a guide to building a low-cost system for growing aquatic plants, and this will be based on the tanks that we built at the Millville Aquatic Research Facility. So thank you very much to all of my funders in this project. And I also want to thank some individuals here. First and foremost, my brilliant and supportive advisor, Dr. Karen Kettenring, as well as my committee members, Drs. Tim Walsworth and Soren Brothers, for their very thoughtful feedback. Starting grad school in a pandemic is weird, but every single one of the people in the Wetland Ecology and Restoration Lab made it a great experience for me. So thank you to all of them. Thanks to Nancy Huntley and Thad Nichols in the Ecology Center, the Watts administrative staff, Melissa Stamp with the Utah Reclamation Mitigation and Conservation Commission, Darren Olson and Casey Williams with BioWest, Nate Norman, who's with the Beaver Ecology and Relocation Center, but helped me so much with the tanks at MARF, the Millville Aquatic Research Facility, Susan Durham with patient assistance on statistic analysis, and last but not least, my friends and family. So thank you, and thank you all for being here today. And with that, I'll take any questions. Oh, and I'm going to turn on the room mics so people on Zoom can hear your questions.
Questions for Kate?
Great.
Awesome talk. Love the plant photos. I was wondering, because these sites are like really at the high end of being altered in preparation for your replanting. I was wondering how important you think that level of site prep is to the success of these replanting techniques? Or do you think that these could scale to places where we can't necessarily like dredge or bulldoze the river channel?
So we don't have a lot of information on how site conditions will affect aquatic plant restoration techniques, but there is some research, particularly out of Florida. So Christine Rojas has a great paper on this, and basically what it shows is that these planting techniques can perform quite differently depending on their environmental conditions. So these results, I think, are going to be pretty specific to sites with similar conditions, but we need to do further experiments to figure out exactly how that all fits in.
If you could speculate on the next step, a place like Provo River Delta or any other large restoration project, are you going to have to plant the whole thing, or can you plant only part of it? And how do these aquatic plants propagate throughout, on their own?
So I'll give you two different points here. The first is that one of the really cool things about the Provo River Delta that I've been able to observe firsthand is that that area was historically the river delta, and a lot of the seeds and oospores of Cara persisted in the sediment. So a lot of the native plants are coming back into the restoration site, which is very cool to see, but that's definitely not the case in every wetland restoration project. So the other point there is that aquatic plants can reestablish very readily through stem fragments. And so you could just restore specific areas, like based on where you're worried erosion will happen. A potential downside of that would be by waiting for those plants to mature and the stem fragments to migrate, invasive species might move in. So it's kind of like a delicate dance of like, if you don't have a lot of resources, you could just plant a few of strategic communities throughout the site, while still having that risk of invasive species.
Thanks.
Thank you. Right.
I'm just curious if you evaluated or have any thoughts on whether different planting techniques might work better or worse in like deeper or shallower places. I know you talked about like the completely exposed sites, but if you have much deeper water, for example, would you still want to do like anchoring on the bottom, or do you think fragments work better? Or just curious about that.
Yeah, so let me think here. So the Christine Rohal's paper out of Florida does look at greater water depths. And anchoring techniques in greater water depths are still effective techniques, but in those scenarios, I think I would suggest using like more anchor, like more weight, larger plants or a heavier weight. For stem fragments, you could still use them in that area, but likely they're going to migrate to shorelines. So yeah, again, delicate dance.
Talking about your fragment experiment, you use that square containment. Do you think you'd have the same response, the same success without that, especially when you think of scaling up in the next steps of the research? Is it even feasible to do like a bigger containment area, if you could just speculate?
Totally. I think for that particular experiment, I think the results would have been pretty similar just due to that water level dropping. So a lot of the plants stayed kind of close to where they were. But if the water levels were different, I think it probably would have differed. And so for me, if I were doing the next step here, I would love to compare, like block off a whole lobe of the pond and compare that to like a treatment in another lobe of the pond. So yeah, I anticipate a larger area would probably perform a bit differently.
We've got a couple Zoom questions too. So the first one is Jimmy Marty asks, are there specific traits of Ruppia that make it more sensitive to planting method? And are there traits that we can generalize to hypothesize the planting methods for other species?
So I'll answer the first part for Ruppia. We only evaluated one species of Ruppia, so I can't say exactly, but I do imagine that there would be differences between species. For example, Ruppia cirrhosa has its flower on this like really cool swirl, and that can mean it moves differently in the water column than other species where it's straight. So like that's a functional difference that I assume would work a little bit differently, but I can't say, that's just speculation. And what was the second part?
The second part was, are there traits that could be generalized just to other species?
Traits that can be generalized to other species. So I think probably, but in order to make that determination, we have to test some more functionally similar species to see if like certain groups, for example, the different species in the genus Stuckenia, if they all behave similarly under these conditions.
We've got more questions on Zoom too. Ronan asked, he said he thought it was interesting that there are a lot of differences between the planting design and methods, and what you expect might happen if you scaled this experiment up with things like different plots in the Intermountain West, different species, all sorts of different things. So I guess, yeah, what would you expect to happen if as you scale up these experiments?
Or maybe just the other wetland area, maybe like Great Salt Lake as an example.
Yeah, let's look at the Great Salt Lake example, because even though it's nearby, there's much higher level of salinity, and a species like Ruppia cirrhosa, for example, does grow well in more saline conditions. So it is pretty site dependent, but I think we still like knowing how to use these planting methods, and if they work in certain environmental areas, we can start to extrapolate based on some site similarities, but probably not too far without further experimentation.
And then we have one last Zoom question from Naomi, who asked, what sort of prep did your field collected samples have before going into the marketplace?
That is a great question. So we worked with UDWR and their aquatic invasive species team to create a protocol to ensure that we're not moving invasive crocules around. And that started at the site itself. So we scoped the site for invasive species. We collected small amounts of plant material because we did not want to impact the native population too much. We brought that back to the lab and made sure those plants were as clean as we could get them with water, then soak them in a potassium permanganate solution to remove any like snails or algal spores or anything like that, and then neutralize that solution. At that point, we introduced it to the tank. So we're trying not to move around anything that we don't want to move around.
Okay, well, let's thank Kate for a great presentation.
Grabs food and you did not sign